In this session, we will study about urine formation and also the mechanism of concentration of the filtrate by countercurrent multiplier system. So first, let's begin with this urine formation. It involves the following steps. One, glomerular filtration, which I also call it ultrafiltration or pressure filtration. And the second step will be reabsorption. So this reabsorption is also called selective tubular reabsorption. Selective tubular reabsorption. See, I told you that when the filtration process takes place, all the useful and also the waste materials will get filtered from the blood, and the body will take whatever that materials that are necessary, okay, back, and that is what we call it the reabsorption. And that reabsorption will be selective process, means only certain substances will be taken back, and for that reason we call it selective tubular. So from the tubules it will be reabsorbed back into the blood circulation. So for that reason we call it a selective tubular reabsorption. And then there will be, okay, secretion. So elimination of some substances from the walls of the tubules, and this is also called selective secretion. I hope this you have understood what you mean by this selective secretion. Again, here also certain substances which are not required, only selected items, that is will be eliminated. So that is selective secretion, or I can also call it simply excretion. Right. So this process I would like to tell you with a simpler diagram. So you'll see here. So this is the Bowman's, okay, capsule, and into this Bowman's capsule, the afferent arteriole, right, and then it comes out as the efferent arteriole. So this is the afferent arteriole and the efferent arteriole, right. So this efferent arteriole, we know it forms the peritubular network. So we can see here it forms a, so I'm just simplifying this, right, and this is the glomerulus, okay. So this is afferent arteriole and this is efferent arteriole, and this is glomerulus, and this is the peritubular network, okay. And we know here the blood is flowing from the afferent arteriole into the glomerulus, where actually the filtration process takes place, and from there, okay, the blood enters into the efferent arteriole, then passes down into this peritubular network. So now let's see. So what this processes, that glomerular filtration, where it takes place, where does this selective tubular reabsorption takes place, and the selective secretion takes place, okay, in this picture. So here, of course, this is the Bowman's capsule. So here the process of filtration takes place. So I'll just mention here as one, and that one I'll mention here, this process is the filtration, right? Then at the next step, that is reabsorption or the selective tubular reabsorption. So from here, that is from these tubules into the peritubular network, and this is, I'll mention as reabsorption. It's reabsorption. Then right from the peritubular network, okay, you can see here the release of certain substances, and this is what we call a the secretion. So that is the third one. So the third one I'll mention here, it is secretion. And then whatever the leftover that passes down is the fourth, that what I call it excretion, right? So this is the place where the actual process, that is filtration, takes place, and from here into the peritubular network is what we call the reabsorption, and back into this okay, the tubule is the secretion, and then whatever the substances that left over in the tubules will be excreted out. Okay, so that process we call it excretion. So now first we will see about this glomerular filtration or ultrafiltration, how it takes place in the Bowman's capsule. Right. So here in the Bowman's capsule, already we know that, okay, so this is the endothelium of a capillary, through which the blood is actually flowing, and below that you have a basement membrane, and then the next layer is the epithelium of Bowman's cup or the Bowman's capsule. So you can see this is the epithelial layer, and the cells of this epithelium are what we call them podocytes. Here I'm not showing their structure, so just I want to mention the layers. Okay, so in between these two layers is the basement membrane. So this is the epithelial cells of Bowman's capsule, and this is the endothelial layer of capillaries. So we know there is a network of capillaries that are formed inside the Bowman's capsule, and in between this you find the basement membrane. So there is a basement membrane, and we know these epithelial cells, right, which are called the podocytes, and their okay, arrangement of cytoplasmic processes, and between those cytoplasmic processes there will be small gaps left, and they are called filtration slits or slit pores. Now when the blood flows, so this is blood flowing through the capillary, and here because of the pressure that is generated, of course, here we can see the afferent arteriole has a bigger diameter, and efferent arteriole has a smaller diameter. The difference in these diameters will actually cause the pressure in the glomerulus, which will help in the filtration of substances from the blood. So here, okay, so certain substances will pass through this, okay, endothelial layer of the capillaries, then basement membrane, and then epithelial cells, and then it enters into the Bowman's cup. So here, already we have seen the structure of this Bowman's capsule in our last sessions. Okay, so this is the Bowman's capsule's space. Okay, we call it a capsular space. Into this, okay, you can see certain substances are getting filtered and they enter into, okay, this Bowman's capsule. So these substances, whatever they have entered, so we call it filtrate. It's the filtrate, or I simply call it the primary urine. Now let us see here how much pressure is actually required, okay, for this process to take place. So that will be the net filtration rate. So I'll see here net effective filtration pressure. So to calculate this net effective filtration pressure, we need to know certain things. One, it is glomerular hydrostatic pressure. So we need to know about glomerular hydrostatic pressure. And so this is the one point. And another one, we need to know about capsular hydrostatic pressure. Capsular hydrostatic pressure is another one. Okay, we should know it. And then one more is the colloidal osmotic pressure. Osmotic pressure. Now let's look at what actually are these: the glomerular hydrostatic pressure and capsular hydrostatic pressure, and also what is this osmotic, that is colloidal osmotic pressure. So first one, let's see what actually is this glomerular hydrostatic pressure. On the glomerular hydrostatic pressure. So this glomerular hydrostatic pressure is the hydrostatic pressure of the blood. It's a hydrostatic pressure of the blood, okay, while flowing in the glomerulus. While flowing in the glomerulus. Okay, right. So it's the hydrostatic pressure of the blood which is actually flowing in the glomerulus, and this glomerular hydrostatic pressure will be around okay 60 mm Hg. Then coming to the capsular hydrostatic pressure, right, here the glomerular hydrostatic pressure will help in this process of okay filtration. And okay, I mean the okay, now glomerular hydrostatic pressure will be counteracted by two pressures. So the two pressures that counteract this glomerular hydrostatic pressure is these two, that is capsular hydrostatic pressure and colloidal osmotic pressure. So that may push the filtrate, okay, back into this okay the glomerulus. So one is capsular hydrostatic pressure and another one is colloidal osmotic pressure. So first we'll see what actually is this capsular hydrostatic pressure. So here the capsular hydrostatic pressure is developed by the fluid in the Bowman's capsule, okay, and also in the renal tubule, okay, that is in the proximal convoluted tubule. Right, so I'll write this here. This capsular hydrostatic pressure, we can also call it the short form CHP. Okay, so that is the capsular hydrostatic pressure, and this is caused because of caused due to already present fluid in the tubules or in the renal tubules. So caused due to already present, right, fluid in the renal tubules that actually pushes back some of these filtered, right, or filtrate back into the glomerulus. Okay, and coming to this colloidal pressure. So colloidal osmotic pressure. So this colloidal osmotic pressure is developed in the glomerular capillaries due to presence of plasma proteins and other solutes, right? So this colloidal osmotic pressure, I'll write it with a short, okay, COP, the colloidal osmotic pressure, I'll just write it. Okay, so this colloidal osmotic pressure is due to presence of plasma proteins and other solutes, okay, in the blood of glomerular capillaries. In the bed of, okay, glomerular capillaries. Okay, right. So as I said that the glomerular hydrostatic pressure is of 60 mmHg, and coming to this capsular hydrostatic pressure, which you can also call it as Bowman's capsular hydrostatic pressure. So this capsular hydrostatic pressure will be of 20 mmHg. And coming to this colloidal osmotic pressure. So the colloidal osmotic pressure is about 30 mmHg. So now let's see how we are going to calculate the net effective filtration pressure. The net effective filtration pressure is, okay, we need to take the glomerular hydrostatic pressure. We need to take this glomerular hydrostatic pressure, okay, minus, okay, as these two are, okay, the opposing forces for this glomerular hydrostatic pressure, so you need to, okay, remove them collectively from this glomerular hydrostatic pressure. So minus the capsular hydrostatic pressure, okay, that is CHP, plus, okay, the colloidal osmotic pressure, that is COP. So we know the glomerular hydrostatic pressure is 60 mmHg. So 60, right, minus, so this CHP, that is capsular hydrostatic pressure, is 20 mmHg, okay, right. So this one and plus, okay, the 30, so you will be getting around, okay, 60 minus 50, that will be, okay, finally get 10 mm Hg. So the 10 mm Hg is the net effective filtration rate. So here the amount of filtrate formed per minute in the kidney, so is what we call it glomerular filtration rate. So the amount of filtrate formed by the kidney per minute is what we call the GFR. So that stands for glomerular filtration rate. It's called glomerular filtration rate. So the amount of filtrate formed by the kidney per minute is what we call GFR. And in a healthy human being or in a healthy adult, okay, person, the GFR will be, so GFR in a healthy man, it will be 125 ml per minute. So if you calculate it for a day, you will get 180 liters per day. So that is 180, okay, liters per day. We will be getting the GFR. So of course, out of all this 180 liters of GFR, all it will not be eliminated out. So we will see that one. So how much of it will be reabsorbed back and how much of that 180 liters per day will be excreted out. Okay, so it will be 180 liters per day. So by taking 125 ml per minute. So next, that's about how the filtration, that is glomerular filtration, takes place, which we also call it ultrafiltration. So almost all the substances except those plasma proteins and okay will not be filtered, and blood cells will also be not filtered, right? And all these substances, whether it is useful or not, so they will get filtered, and that forms the primary filtrate in the renal tubules. So now from this filtrate, the useful substances will be reabsorbed back. So that's, we'll see, is called selective tubular reabsorption. So the next one is, that's the second step. It's called selective tubular reabsorption. So here in the selective tubular reabsorption, okay, which is the second step, as I told, in this what happens, 99% of the filtrate will be reabsorbed by the renal tubule. So 99% of filtrate is reabsorbed. Okay, so it is reabsorbed by the renal tubules. So here this reabsorption process will take place in different segments of the nephron, and this reabsorption process, so the reabsorption will be either okay active process or by the passive process, right? We know already the active process, which means it uses energy to take up those substances, and passive process without using the energy by means of diffusion, okay, or by okay facilitated diffusion, whatever. So through that, if it takes okay or reabsorbs those substances, we call it passive process. So here we'll see the substances like the glucose, which is filtered, okay, then amino acids which are also filtered and they are present in the filtrate. Now some of these will be reabsorbed based on the requirement of the body, and also the sodium ions which are present in the filtrate. So these are present in the filtrate will be reabsorbed by active process. So they are reabsorbed by, okay, active process. And some of these nitrogenous wastes will be also reabsorbed back, okay, to maintain the osmolarity, right? That will be absorbed by a passive process, okay. And this selective reabsorption takes place in the PCT and also in Henle's loop. So selective reabsorption takes place in, okay, the PCT, right, Henle's loop, and of course also in DCT. And okay, DCT. So in all most all the parts of the nephron you will see the selective reabsorption taking place. Already this we have seen in our earlier sessions where I have discussed the functions of the tubules. One and the third step in the formation of urine is the secretion, that is, okay, selective secretion, so which we can also call it excretion, or one more word we have here is augmentation. Excretion or augmentation. So this will be the final step in the urine formation, and okay, right, this will take place in occurs in DCT, that is distal convoluted tubule. Substances like the hydrogen, okay, and also the potassium and also ammonia. So these substances will be secreted, that is selectively secreted, and that actually helps in maintaining the pH and also the sodium potassium levels in the blood circulation. Okay, so that we have already seen in the functions of a distal convoluted tubule. That's about the urine formation. Now we will see the mechanism of concentration of the filtrate. Concentration of filtrate. And this we will see with the countercurrent multiplier system. So here the mammals actually have that ability to produce concentrated urine, and when we see the outer cortex to inner medulla, okay, say this is the cortex and this is medulla, right, we see there is a increase in the osmolarity from the cortex to the medulla. So I told you that there are fluids that fill up these medullary regions, and we call it as interstitial fluid, so that will surround the nephrons. So in fact I say that the Henle's loop will be dipped in the interstitial fluid, okay, and uh, from this cortex towards the inner medulla. So we have outer and inner medulla, and that will be almost to the the tip of the renal pyramid. So almost to the tip of the renal pyramid, from outer cortex to the innermost medullary region. So here this is the tip, and we know here lot many collecting tubules finally open into the minor calyces. So minor calyx. Okay, so here you will find there is more osmolarity. So here we have a very high osmolarity. So we know the meaning of osmolarity is the concentration of solutes. So more the solutes, more will be the osmolarity. This osmolarity is mainly caused by two substances. That one is sodium chloride and another one is urea. Okay, so I told that from the cortex to the inner medulla there is an increasing osmolarity. That increasing osmolarity is due to or caused by, so increasing osmolarity towards inner medulla is caused by mainly the two substances. So that is a sodium chloride and another one is urea. So in the interstitial fluid you will find more quantity of this sodium chloride and urea. This actually helps in concentrating the urine by absorbing more amount of water from the tubules into the interstitial fluid. So how it is seen and how it actually helps in concentrating the urine. I told that loop of Henle plays a very important role in concentration or in concentrating the urine, and this along with the vasa recta. So a part of peritubular network, that the blood vessels which run parallel to this Henle's loop will play an important role in concentration of the urine. That we will see how it occurs. So let's see this counter current multiplier system. Okay, so here you see this is the cortex region, and this is the outer medulla region, and this will be the inner medulla region, right? And I told you that from the cortex towards the inner medulla, the concentration of interstitial fluid will be increasing. Okay, so that will be from 300 milliosmoles per liter. Okay, so I'll mention on this side. So here it is 300 milliosmoles per liter. So from the 300 milliosmoles per liter, it will increase to 1200 milliosmoles per liter. Okay, so this will gradually increase. You will see around 600 milliosmoles from 300 in the outer medulla region. So here you may find 600 milliosmoles per liter, and then increasing to 900 milliosmoles. So here to 900 milliosmoles per liter, and finally to the deeper part 1200 milliosmoles per liter. If we one point here, when we see the desert animals or the animals that live in dry conditions with less amount of water available, okay, So in them, the kidneys will have more number of juxtamedullary nephrons. So we have seen in our earlier sessions that juxtamedullary nephrons will have very long loop of Henleys. They also show the thin and thick segments of the ascending limb, whereas the thick limb, I mean the thick segment is not present in the case of cortical nephrons. So if we find in case of human beings, I already gave you that the percentage of cortical and juxtamedullary nephrons. We have more number of cortical nephrons as we don't live in very much dry conditions. The juxtamedullary nephrons are around only 15%, whereas the cortical nephrons are about 85%. So in case of desert animals, the number, that is juxtamedullary nephrons number, will be more when compared to the cortical nephrons. Like in the North American kangaroo rat, you will find more number of juxtamedullary nephrons. Okay, fine. So we understood that the Henle's loop is mainly meant for concentrating the urine, and here some substances that pass out from the renal tubules, they enter into this interstitial fluid. So here all it is surrounded by the interstitial fluid, and I'm telling about the osmolarity of the interstitial fluid. So from the cortex to the inner medulla, increasing from 300 milliosmoles per liter to 1200 milliosmoles per liter. Okay, so this MOSMOL, that stands for milliosmoles. Okay, now this is the vasa recta. Okay, and the vasa recta, right, is present. So same thing, actually it is present here, that I have kept it aside. So this one you have to see it here. Okay, so in this case you will see in the renal tubules the flow of the filtrate. So this is the direction. In the descending limb, the filtrate moves down, and in the ascending limb the filtrate moves up. So here these two are in countercurrent, and in the same time here you will find the blood in the vasa recta will be flowing in this direction, and here also the flow of blood is in countercurrent. Okay, right. So this is the one that comes from the efferent arteriole. So this is from the efferent arteriole. We know the efferent arteriole will make a peritubular network on this PCT, Henle's loop, and DCT. So I have just we made a simpler one. Okay, actually this should be present here, that I have drawn it separately. Clear? So this is what we call it the countercurrent. So in these two limbs, the ascending and descending limb of loop of Henle, the filtrate is in countercurrent, and the same time in the vasa recta also the blood is in countercurrent. Now how this actually helps in concentration of urine. Okay, so here we know that this descending limb of loop of Henle is permeable to water and impermeable to solutes. Okay, so here actually the water will come out, but solutes will not come out of this. So as the water comes out, the concentration of the solutes will increase, and this will increase from the PCT to the hairpin of Henle's loop. So this is the point, the bend, the hairpin of the Henle's loop. So by the time the filtrate comes here, the concentration of the filtrate will be almost equal to the concentration in the inner medulla. So here inside this you will find 1200, okay, milliosmoles per liter. I'm not writing here, just I'm I'm just written the number. So actually it is 1200 milliosmoles per liter, and this will go on increasing from this point. So if you see here, it is around, okay, 300 milliosmoles per liter, and as it comes down, okay, it becomes 400 milliosmoles. So you can see the water is actually coming out in this case. Now here we know in the ascending limb, which actually has what you call the movement of NaCl, but it is impermeable to water. So NaCl will come out from the ascending limb, right? So if this NaCl comes into the ascending limb, so it increases the osmolarity in the interstitial fluid, okay? And for that, from the descending limb, the water will come out, okay. And with this mechanism, what happens as the filtrate moves down, it will be from 300 to, okay, 400, then a little bit deeper it will become 600 milliosmoles per liter, then it becomes 800, then 1000, and then finally it becomes, okay, 1200 milliosmoles per liter in the filtrate, okay. Now as it moves up again, the filtrate will get, okay, diluted. So again from here it will be 1000, then okay 800, then it becomes, okay, 600 and 400, and then, okay, by the time the filtrate reaches the distal columnar tubule, it will be back to the same, okay, concentration as that of the interstitial fluid in the cortical region. And some of the NaCl that is released into the interstitial fluid, okay, will enter into the vasa recta. So here NaCl, and some, okay, water will also come out from the vasa recta into the interstitial fluid. Corresponding to this, you will see the same osmolarity even in the case of the vasa recta here. So you may find, okay, so here 300 milliosmoles, then coming down 400, and again, okay, 600, and then 1000, okay, and here to 1200, and back again, you may find, okay, the same, like, okay, 600, and again back to 400, and then to 300 milliosmoles. So some, okay, portion of NaCl will also come out from the ascending limb of the vasa recta. So this is the ascending limb of the vasa recta, and this is the descending limb of the vasa recta here, and this is the descending limb of loop of Henle, and this is the ascending limb of loop of Henle. So here you will see some portion of sodium chloride coming out, and that may again enter into the descending limb of the vasa recta, right? From the, okay, the collecting duct. So this is the collecting duct. From the collecting duct, so taking out, I mean, the pass out of water into the interstitial fluid, and some portion of urea also comes out from this collecting duct and enters into the interstitial fluid, and some of that may again enter into this, okay, Henle's loop, right? Now the point is here, this mechanism actually helps in maintaining the concentration gradient in the medullary uh, interstitium. So when we maintain this concentration gradient in the medullary interstitium, so what happens is it allows the reabsorption of water into the interstitial fluid. Okay, and some of this will again enter back into the blood circulation. Okay, right. Then so here because of this, okay, there will be even, I mean, the as the filtrate moves up, we will see here there is a concentration, I mean the filtrate becoming concentrated here. Okay, and finally the I mean after the whole process is completed, okay, the kidneys will be able to produce urine that will be 4 times concentrated than the initial filtrate. So this is the location where the, okay, filtration uh, okay, filtrate first formed here, that is initial filtrate. Okay, so by the time it reaches into the collecting duct, okay, it will be 4 times concentrated than with the initial initial filtrate concentration. Here the only thing that we see here in the countercurrent multiplier system is to maintain the concentration gradient in the medullary interstitium. Okay, so by this sodium chloride and the release of urea, you can see the sodium chloride, okay, entering into the interstitium and also urea into the interstitium, and this, okay, mechanism will help in concentrating the urine as it passes through the, okay, tubules. So that's the only thing why this countercurrent multiplier system is present in kidneys of mammals. Right, so with this I'll end up, and in the next session we'll see about regulation of kidney function and maturation.